FAS-T bioenzyme is a product-content enzyme. Our aim for this study is to produce enzyme that helps to transform large-scale waste into high-quality compost started with microbes. The problem now is that abundance of empty fruit bunch (EFB) was found in Malaysia that contribute 23% of total palm oil waste, which is equivalent to 21.36 million tons. So, the solution is we use EFB in our bioenzyme because it contains microbes that can work well in high temperature, range from 40 to 80 degrees Celsius. This is why our bioenzyme functioning best because it contains thermophilic bacteria to work in composting process. Thermophilic bacteria helps to break down the waste further, and the higher temperature acts as an ideal environment for the breaking down of complex compound in the waste. For the methodology part, this research started with isolations of bacteria from EFB. The procedure included with media preparations, isolation process, and the media incubated for 24 to 48 hours at different temperature to test for its thermotolerance. This is the result shown from the colony counts where multiple colonies were formed after incubation period. The data indicates that microbiome from EFB exhibited high microbiome activities during thermophilic phase. Continue with enzyme analysis, the selected isolated bacteria were screened for amylase, cellulase, and lignin peroxidase. The amylase activities was characterized by the formations of clear zone on the media which function as energy source for growth. Cellulase activity is determined by the formations of clear zone diameter. Lastly, the discolorations of methylene blue was observed to screen for lignin peroxidase activities. These observations show the positive growth of enzyme from isolated bacteria. Lastly, to test our bioenzyme performance, we have conducted tests on a lab-scale composter for 24 hours. The bioenzyme is added at the early stage of composting process. The nutritive content analysis showed the quantitative measure for nitrogen, phosphorus, and potassium with 1,459.23 mg per kilogram, 1,096.16 mg per kilogram, and 2,188.85 mg per kilogram respectively. In conclusion, the novelty of our research is FAS enzyme contain microbes that are able to break down organic waste at high temperature. It is new finding of local product that contains thermotolerant strain bacteria from EFB which can enhance the nutritive contents of compost and its quality. Besides, it has potential to be used in agriculture industry and helps to overcome the abundance of EFB waste with the approachable method and environmental friendly.